Yeah, today we are going to discuss about streptococcus. Yeah, streptococcus, that is the cocky with the streptosep. And the general introduction for the streptococci is first it is the gram positive, like the staphylococcus, catalase negative, but the staphylococcus were also some of the gram positive and catalase positive and catalase negative too. But streptococcus all are the catalase negative. These are the not non-motile cocci arranged in the chain form in the one axis. So it is called the streptococcus. Yeah, due to its shape or due to its size, we can tell it's the streptococcus, and it required the blood or the serum reached. A serum reached media for the normal growth because it need high quantity of the nutrient for its growth, and it is found as the normal microbial flora in the human being in the upper respiratory tract and animal, but exception is the streptococcus pyogenes, as it is pathogenic in the nature. Discovery of the streptococcus means there is the name of the some scientist who's make their contribution in order to discover about the streptococcus. First is the Billroth. Yeah, Billroth means he seen and described about the streptococcus and gave the name and after that there is the Augustine, and his contribution is to differentiate how can we differentiate streptococcus and staphylococcus. He gave the difference between the streptococcus and the staphylococcus. Then another scientist is Rosenbach. Means he gave the name, just only give the name to the streptococcus pyogenes. Yeah, here is the streptococcus pyogen. And then the classification of the streptococcus, it is based on the oxygen. Yeah, oxygen, either it can use the oxygen or not use the oxygens, that is the facultative anaerobe and the obligate anaerobe. Means as we go in the facultative, there is the base of the okay, based on the hemolysis activity, the facultative anaerobe anaerobe streptococcus we can classify into the three groups. First is the alpha, means the partial hemolysis, or some of the heme heme protein are lysed or the breakdown from the blood, and some are remaining there as it is. And there is the beta hemolysis. Then beta hemolysis means here we can just simply think beta means complete. Yeah, complete hemolysis means all whatever amount of the blood is present there, they must be hemolyzed, or the heme protein should be away or break down from the blood. And there is the gamma. Yeah, there is the gamma, and the gamma means there is no any kind of hemolysis takes place, or the no blood is hemolyzed, or the breaking down means no heme protein is out of that. And then the beta hemolysis. Yeah, beta hemolysis is again divided into the 20 classes or the 20 groups based on the serological division, or the serology means whatever the serum is present there inside that one. And they are the A, B, C, D, E, F, G, H. Then I and J are left, or the except this up to U and P. Group A, or Group A streptococcus, as it is simply called as the gas streptococcus. A gas streptococcus, they are divided into the 80 classes based on the protein present on their surface, or the surface cell. Thus, means the cell wall. Yeah, hemolytic grouping of streptococcus are alpha, beta, and the gamma. Yeah, for the grouping. Then alpha hemolytic streptococcus. In this, we have already discussed that they are the partial hemolysis takes place here, and in part they show greenish discoloration. Means partial hemolysis with the greenish discoloration is there, and zone of lysis is 1 to 2 micrometer. Yeah, or 1 to 2 micrometer is the zone of lysis. How much there is the breakdown of the heme or the blood is the zone or the area for the breakdown of the blood, that is the 1 to 2 micrometer. An example of the some alpha hemolytic streptococcus are streptococcus viridans, means like the polysaccharide capsule. Opportunistic in nature means whenever our immunity power become weak, they can attack. And streptococcus pneumoniae are the causative agent for the pneumonia. Then second is the beta hemolytic streptococcus. Here the produce the sharp defined clear color zone that is about 2 to 4 micrometer, and complete hemolysis takes place and is responsible for the majority of the streptococcal diseases. Means maximum streptococcal diseases are caused by the beta hemolytic of the complete hemolytic streptococcus. Then third class is the gamma hemolytic streptococcus. In this we can think that gamma means no, means the gamma hemolytic means there is no change in the color of in the blood agar, and no hemolysis takes place. An example are the streptococcus facialis and enterococcus are the two best examples for the gamma hemolytic streptococcus. Then serological grouping by the Rebecca Lansfield. Lansfield is the name of the scientist which give the serological grouping of the beta hemolytic streptococcus into the 20 class. We have already discussed these things. And based on the nature of the carbohydrate with the C antigen, he classified the streptococcus into the 20 specific species based on the presence of the peptidoglycan layer. That is A B C D E F G H, then I J R exception, and K L M N O P Q R S T U V, not WXYZ. And gas means the group A streptococcus means majority of the infection, yeah, majority of the infection are caused by the streptococcus, and especially that is the streptococcus pyogen. The special name is the streptococcus pyogen. It is the main causative agent for the streptococcal disease or the infections. Then Griffith classification of the group A streptococcus. They were means he classified Griffith classified or divided the group A streptococcus into the 100 different serotype based on the present of the protein on the cell wall, that is MTR protein. Then here the diagram where we can see some things. That is here is the M protein. M protein means it is the main causative of a protein which can infect the normal human bodies, or that is the one of the toxin or act as the toxin whenever it come attached to the animal or the human bodies. And lipoic acid, this is also, yeah, it's also necessary things which is in the capsule with the outer covering and then the cell wall is just inside the capsule. And plasma membrane as is in M protein, lipoic acid, protein F and SPF in vagin. These are the some kind of in vagin streptolysin. These are the toxic substances. Streptokinase and proteases. These are enzyme and exotoxin SPE ABC. These are the toxic substances inside the streptococcus. And then the pathogenicity. Yeah, in the pathogenicity we will discuss about the first is the capsule, that is the outermost layer of the cell of the sum of the beta hemolytic streptococcus, and mostly present in the what, group A streptococcus. And in the in vitro culture the capsule production is on the logarithmic phase of the growth but destroyed due to the production of the hyaluronidase. It is also enzyme which is also present in the human bodies and which can attach or which make the specific receptor for attaching of the hyaluronic acid, which is also already present in the capsule of the streptococcus and give the mucoid colony an phagocytic in nature. Means it is antiphagocytic, means it can inhibit, yeah, it can inhibit the phagocytis inside the human bodies. And capsule help to bind with the CD44 cell molecule receptor present in the pharynx with the hyaluronic acid or the acid of the acid binding proteins. Then another is type specific protein antigen, that is the M protein. Yeah, and the M protein we will discuss about it is the major virulent factor or the major causative factor which can affect the human bodies, and it is already found in the cell surface. Antigen is cell surface, yeah, and the composed of the two polypeptide polypeptide chain complexed in the alpha helix. Means polypeptide chain is the major things for this, and there is a resist to the acid, yeah, resist to acid and the heat and susceptible to the tryptic digestion and soluble in the alcohol. As resist to acid and heat means there is no any kind of effect from the acid using acid or the HCl or the H2SO4 or the heating the that one due to the M protein. Yeah, M protein is also able to resist the acid and the heat, and it also make the streptococcus soluble in the alcohol so that we cannot hear it and inhibit the phagocytosis. Yeah, inhibit phagocytosis means due to the M protein present in this, there is no any kind of effect from the WBC cells, yeah, white blood cells, other any kind of phagocyte cell, and monocyte lymphocyte cells, and activity by inhibiting the opsonization through the alternate complete pathway. There is the alternate complete pathway we will discuss in the further lecture. Through the use of this alternate complete pathway, there is the phagocytosis inhibitants due to presence of the M protein, and it is also bind to the fibrinogen, which together bind to the beta 2 integrin of the neutrophil, leading to released. Yes, up to here we have completed, and further lecture the further type specific protein antigen in culture of this and resistance capacity. Other things we will discuss in tomorrow. So thanks for this much.